Okay, lab 5, we will do a hands-on exercise on plasmid DNA isolation. Especially, we will try to isolate plasmid DNA called PBL322 from E. coli cells that are grown overnight. Those E. coli cells carry this plasmid because they were transformed before. Okay. I guess biotransformation lab results, I guess if this lab is done after the biotransformation lab, I guess you need to check the result by looking at the plates and counting the colonies, transformed colonies, and calculating the transformation efficiency. So that's what it means. Okay, so what is plasmid DNA? So I'm hoping you probably would have done your lecture already. The lecture, probably it's 5, that talks about the genomic structure of prokaryotic cells and eukaryotic cells. When it talks about the genomic structure of the prokaryotic cell, it talks about circular chromosomal DNA as well as the presence of plasmid DNA inside of the nucleoid area. So when you talk about plasmid DNA, those are what? Independent DNA that are self-replicating, and then they may carry some important genes, so we call them extra chromosomal DNA. Okay, so and the plasmid DNA are circular as well. Okay, so they are naturally found plasmid DNA very small compared to the chromosomal DNA, and that they are replicated independently outside of the chromosomal DNA. And what do they do? Naturally, plasmid DNA molecules can be transferred from one cell to another. There are different ways of doing that. One well-known mechanism for plasmid DNA transfer is conjugation between two bacterial cells. There may be a development of conjugation tube. Through that conjugation tube, cells can transfer some copies of plasmid, and then if those plasmid DNA carries certain genes that are beneficial for the cells, such as maybe resistant to antibiotics, or maybe the genes that carry toxins so they can fight against other competitors among themselves, so that those will give the benefits for the cells to survive better. Okay. And structure, I already told you, small circular DNA sources, because they are transferred from one cell to another. That's where they come from. Once they are taken up by a cell, they can stay there and then they can be replicated. Depends on the copy number of plasmid. There may be tens of copies, there may be hundreds of copies per single cell. So there are multiple copies normally found from a single cell, copies of plasmid DNA. Okay. Oh, the last question, the importance in biotechnology. So why is that important, the plasmid DNA? Okay, so the reason is, plasmid DNA molecules are used as a vector or delivery vehicle. Okay, when you transfer or deliver a new gene of your interest into a host cell, such as bacterial cell, you use these plasmid DNA molecules as a delivery vehicle. The reason is, a gene that's existing in maybe double-strand DNA cannot be readily delivered into a cell, because when it's delivered as just a linear DNA, it can be degraded by DNase enzymes that are found from cells very quickly. So instead of delivering as linear DNA, you want to deliver your gene inside of the plasmid DNA, so as a kind of recombinant form. Recombinant meaning your gene of interest is recombined, added to the plasmid DNA, so it gives you a full circle. And then once that plasmid DNA, maybe if just a single copy of it is delivered into the cell, it can be maintained because plasmid DNA can be replicated once it's taken up by the cell. So it's a very efficient delivery vehicle. So since you can easily deliver your gene of interest to different bacterial host cells, once those cells have taken up your gene, then you can use those cells, those recombinant cells, as basically a platform or factory that can maybe maintain your DNA, as well as sometimes it can work as a factory that can maybe transcribe and translate the product out of your gene. So what you are interested in is a product, which is maybe RNA or protein out of that gene. You can trigger the cells to transcribe and translate the gene of your interest, so you can get the final product made. And those cells bacterial cells can be easily duplicated if you grow them, with the fresh medium, food, oxygen, and all those things. They can easily grow quickly. It's not very expensive. So it's very easy to maintain those cells and maybe duplicate the gene copies or duplicate the products out of your gene. So plasmid DNA is very important for that purpose. Okay. What you're going to do is a small-scale isolation of plasmid DNA. So what does that mean? Small scale means small size. You are trying to isolate the gene. Okay. The plasmid DNA. So small scale is normally about 1 ml, 2 ml, 3 ml, 4 milliliter volume quantity, but large scale we consider as what, maybe tens of liters or hundreds of liters or thousands of liters are large scale. Okay, so small scale is kind of a bench scale. So if you are maybe growing cells overnight, maybe test tube, which may hold about 4 mils and 5 mils. Okay, then out of that 4 mil, 5 mil, you can use about 1 milliliter of overnight grown culture, which will have a lot of cells, because the cells will be very dense in that culture because they grow not overnight. And if you look at the growth curve, they may be in the stationary phase. Okay, and then you're going to harvest the cells. Hopefully those cells all have the plasmid DNA copies, and then from those cells you're going to isolate the plasmid DNA out. Now, how do you do that? You'll collect the cells using centrifuge, and then you will treat the cells with some different chemicals to break open the cells, and you'll release what the cells have inside. They may have, chromosomal DNA, plasmid DNA, RNA, whole kinds of other proteins and enzymes, all those things, and you'll sort out those molecules, okay, like protein, lipids, and chromosomal DNA, and using different ways, different methods like centrifugation or enzymatic treatments, and you'll try to select for the plasmid DNA you want. Okay, so typically from 1 mil of overnight grown culture that's in stationary phase, you can isolate about, if you are really good at it, 2 to 5 microgram of plasmid DNA. So that's a typical small scale plasmid DNA isolation yield. So the plasmid DNA you'll be dealing with is called BIOL221Lab5-1. Normally, it's a nomenclature of the plasmid DNA name. So normally you start out with small p. Small p stands for plasmid DNA, and then BIOL221. Normally, the next two letters in English alphabet may have something about where that plasmid DNA is made. 322 maybe, I don't know, maybe size or whatever the number of plasmid DNA that's made by that, that place. Okay, so yeah, so that's the name. So the map of this DNA, okay, is right here, plasmid DNA BIOL221. The entire size is about 4,361 base pair. So it's not that big, so that's a size. And within the circular DNA form, there are some functional genes that are found. Okay, so if you look at that part from here to here is called BLA. It's a beta-lactamase gene, which gives the antibiotic, the ampicillin resistance. If this gene is, transcribed or translated within a bacterial cell, that will give the ampicillin resistance. We talked about ampicillin resistant gene in the previous lab, okay? And then it also carries another antibiotic resistant gene, that's a tetracycline resistant gene right there. Okay, so tetracycline resistant. So it carries two different antibiotic resistant genes. So why do you want to do that? Because these genes give some selection strategies when you clone your gene within this BIOL221 and deliver into a host cell, the bacterial cell. Selection strategy means once E. coli cells are transformed, okay, they will give you what? Antibiotic resistant. Okay, the cells that have taken up this BIOL221 will be able to survive with the presence of ampicillin as well as maybe tetracycline, right? So the cells that were not transformed will not be able to survive. So that's what selection means. Okay, and then So REP may be replication origin. So for this plasmid DNA to be replicated and maintained inside of a cell, you have to have a replication origin where the DNA replication starts. So, and then you see all these marks and names, those are recession enzyme cutting sites. So recession enzymes are what? Endonuclease enzymes that can recognize specific DNA sequence and cut. Cutting means like cutting the phosphodiester bond. So you have so many of these enzyme cutting sites because you can use some of those enzyme cutting sites to open up this plasmid DNA, and then you can insert your gene of interest in there. Okay, so by doing so, you can use this plasmid DNA as your delivery vehicle, the vector. Okay. So the cells that containing this BIOL221 should have inoculated. Inoculated meaning transferred into a fresh medium aseptically, so nothing else is growing except for this cell. So we use Loria broth, that's a growth medium, with the ampicillin. So because only the cells that have taken up BIOL221 will be growing, so that's a selection strategy, nothing else. Okay, so probably that was done probably the day before you come into the lab, and then they've been growing on a 37-degree Shaker incubator. So 37 degrees, same temperature as our body temperature, because normally E. coli cells are originally found from the gut of animals and humans. Okay, so our human body temperature is a perfect temperature for them to grow. Overnight, well, it's a long time because they double every 20 minutes under the optimum temperature, so they should have doubled so many times, and there should be gazillions of cells in there. And why do you have to shake? You normally grow E. coli cells in the Shaker incubator. By shaking, you can disturb the cells, so cells can be continuously suspended. Suspended means what? They are floating in the liquid. Okay, if you don't shake, what's going to happen to cells will settle down at the bottom. Then what's wrong with that? If cells are settling down at the bottom of the tube, there may be cells that are crushed at the bottom. Layers and layers of cells will be found. So the cells at the bottom, okay, will be depleted from oxygen, depleted from nutrients. So they'll die from it. So that's not good. So that's why you want to suspend and shake your growth container. Maybe it could be a test tube, it could be a flask, or it could be a bioreactor, whatever you use. Okay, so shaking, suspending the cell is very important. Okay, and plasmid mini preparation. So that's plasmid DNA isolation procedure. Mini means small scale preparation, preparing procedure step by step. Number one step, you're going to transfer the 1.5 ml overnight cell culture volume into a microcentrifuge. A microcentrifuge tube is a plastic tube that can hold about 1.5 ml total volume. Okay, you can use it and throw away, disposable. Okay. And then once you transfer them using your micropipetter, okay, you'll be probably familiar with micropipetter by now. So set your micropipetter, maybe like 750 microliter, and pipette it twice. Then you can get 1.5 ml, which is 1500 microliter. Then the microcentrifuge tubes will be closed by placing the lid back on, and then you're going to place them in a centrifuge, tabletop microcentrifuge that you will find from your bench. You will spin the centrifuge about 1 minute at the maximum speed. So 1 minute is not a long time, but by spinning quickly, you will create centrifugal force that will be applied to the cells that are suspending within your liquid culture. So since cells are heavier than your liquid medium, they will come down as a pellet at the bottom of the tube. That's how you can separate the cells from the liquid medium. So you can collect the cells only from the medium. Okay, and then once that's done, stop the centrifuge and recover your tubes. And then once you have recovered your tube, you need to discard the medium, the liquid, clear liquid floating on the top of the tube using an aspirator. Aspirator is located in the lab. It's basically a vacuum pull trap device that we made in-house. So you can use that to get rid of the liquid medium without disturbing the pellet at the bottom. Okay, after separating your cells from liquid medium, I'm going to stop right here.